Hello, my name is Jennifer Ison, and I'm going to be talking to you today about how bee visits impact a plant's male fitness. I want to start by highlighting the co-authors on this project. So I teach at the College of Worcester, which is a liberal arts school, undergrad only in Northeast Ohio, and these are all the student researchers who've worked on this project. We have Evan, Mia, and Zeke, who did a lot of the field work back in 2018, and then seed processing, and then even some of the lab work as well. We have Mayana, Ren, and Avery here who grew up the seeds and tissue sampled them and then did a lot of the lab work. And then finally, Lauren and Sarah, who finished up the lab work this last year, even with all the COVID procedures. Here, so what do we study? Well, we are interested in plant reproduction and specifically how pollinators impact a plant's reproduction. So most plants, 90% of plants, are animal pollinated. Most of those are insect pollinated and most of those are bee pollinated. But plants are usually visited by a number of different floral visitors, and these visitors can vary in their pollinator effectiveness. And we're going to define pollinator effectiveness as a per visit contribution to a plant's reproductive fitness. One such plant that has a number of different floral visitors is our study plant, Echinacea angustifolia, which is visited by mostly native solitary bees. Echinacea we use to study the prairie because we think it has traits similar to many prairie plants, including long-lived, self-incompatible, insect-pollinated, and pollen-limited, especially in small isolated remnant populations. We study this plant in west central Minnesota in these populations, like here, that are mostly along the roadside surrounded by farm fields. Now, before I get into the research, I just want to acknowledge that this work takes place in the homeland of the Eastern Dakota people who were violently removed in the 1860s and not far from the present-day Upper Sioux community in Granite Falls. What the Echinacea Project studies is prairie fragmentation and destruction, which is a direct consequence of colonization, including the forceful removal and genocide of the Eastern Dakota people. There is also a long-standing history of systemic oppression and government assimilation policies. According to the Dakota-led rural nonprofit, Dakota Wichohan, there are about 4,000 Dakota people in Minnesota, but only an estimated 8 fluent Dakota speakers. This nonprofit aims to preserve Dakota as a living language and transmit the Dakota way of life to future generations. And they do this through a number of really neat youth programming. They have curriculum for teachers and lessons for teachers. So I put links to this nonprofit in the chat, and I encourage you to support it. I also, since many of you are educators like me, I also wanted to take this time to highlight a few resources I saw on Twitter a couple of weeks ago. These are resources created by some early career folks to diversifying and decolonizing your syllabi, and I put links to these resources in the chat as well. I know I've already started thinking about fall planting, and these resources have already come in very handy. So now that I've showed you a little bit of the landscape of the area, I want to tell you a little bit more about the pollinator community. So I mentioned that Echinacea is visited by a number of different native bees. These are mostly solitary ground nesting bees, including Melisodes, Andrena, a number of different bees in the Halictidae, including some sweat bees. And they're mostly solitary, some are semi-social or eusocial. But the interesting thing is what we don't see visit Echinacea is bumblebees and honeybees. They are around in the area, but they just don't seem to visit Echinacea. And so I think this makes a unique system to study these other bees and look at how effective these other bees are without these two groups of species that are more widely studied. So we asked the question, do Echinacea's pollinators vary in their pollinator effectiveness? So we've done some previous work on pollinator efficiency in Echinacea. Back in 2010, 12, 13, and 14, we let pollinators visit unvisited Echinacea heads and videotape them, and also assessed how effective that visit was. Here is Maureen doing that. You can watch all these videos on the Echinacea Project YouTube page. All right, so here are the results from that study, which we published in AJB in 2019 with Maureen as the lead author there. And so what we have on our x-axis is receptive styles at time of visit, and on our y-axis, we have proportion of styles that shriveled, and that is an indication of successful pollination. So the higher the value, the more successful the pollination. And each of the dots, what they indicate, or the points, the different colors and shape indicate a different pollinator group. And so what I hope you notice is that most of the points that are higher are these gray circles, and that is Andrena helianthiformis. And Andrena helianthiformis is a specialist in our system, and she was significantly more effective than the other bees in our system. So which are mostly all down here. So what we concluded is that per visit, Andrena helianthiformis is a significantly more effective pollinator than other bees, but I added some asterisks that said for female fitness, because this is really just a measure of seed set. She was more successful at setting seed per visit, but we don't know how effective she is at siring offspring on other plants. And that is key, because when we think about our definition of pollinator effectiveness, we said contribution to a plant's reproductive fitness. So both its ability to set seed, but then also its ability to sire seeds on other plants. So how can you quantify pollinator effectiveness for male fitness? Well, there's a few things that have been done. One is looking at pollen removal, so per visit, how much pollen is removed. The issue there is that most of the bees in our system are actively collecting pollen to partition to their offspring. They make little pollen balls and lay an egg on top of them. And we know that this can actually cause pollen depletion, because we want the plant wants the pollen removed and then deposited on another plant. In fact, in another system with a specialist Andrena here in Spring Beauty, Allison Parker and collaborators found that the specialist Andrena in that system was so good at collecting pollen that she depleted it from the population and was an inefficient pollinator. So another way you could look at it is looking at the ratio of pollen removal to pollen deposition on a stigma, because usually the more pollen that's deposited, the more seeds that are set in that fruit. The problem here is that echinacea, as in other composites, is uniovulate. So in theory, just one pollen grain needs to land on the stigmatic surface to fertilize the ovule. And we did look if there's any correlation with the number of pollen grains on the stigma and whether that ovule gets fertilized, and that's what Mia did back in 2018. She did a bunch of hand crosses, and the answer is no. So both of these are not really good measures of male fitness, at least in echinacea and probably other composites as well. So the reality is that following individual pollen grades is hard, but we have this experimental plot where many of the echinacea have been genotyped at 10 microsatellite loci. So if we could just know which kind of bee move the pollen that sired a given seed, we have the molecular tools to know which plant is the sire. So how do we figure out which bee moved the pollen? So these fine people here are strategically set up in this experimental plot to monitor flowering. And what we did is we made echinacea functionally dioecious and then restricted pollinator visitation. And so what I mean here is that we assigned most of these plants to be male plants. And then what would happen is we would have a single visit come. So let's say an Andrena visit came to this plant. From then on out, this sire plant is now an Andrena plant, and only other Andrena bees are allowed to visit this plant. And then we had other sire plants where if, say, an agapostumum came, from then on out, only agapostumum were allowed to visit this plant, and other pollinators were shooed away. That's what they're doing. They're standing in the field, counting visits by our target pollinator group and shooing away other pollinators. And then we had a set of plants that were maternal plants, and they could be pollinated and they could be visited by any of the bees. We did this in 2018 in 5 days. We had 316 plant sire day combinations, over 1,000 visits to our sire plants, and 64 open pollinated maternal plants. So then we collected the seed heads and we brought them back into the lab, and we individually removed the fruits from the maternal plants. We grew and tissue sampled the offspring. We had 404 resulting offspring, and then we extracted DNA and genotyped them at our 10 microsatellite loci and assigned paternity using service. So the key is once we assigned paternity to the offspring, we know some really critical things. We know which pollinator taxon moved the pollen that sired that offspring. We also know the number of pollinator visits to that sire plant there. So what did we, and this then gets us our per visit siring success. So what did we find? Well, so here is the result of all of that hard work. On our x-axis, we have the 6 pollinator groups that we had enough samples from, and this is number of offspring sired per pollinator visit. So what this means here, so take this point right here. This is a small black bee, probably a dialectus. So one visit by this dialectus to the sire plant resulted in 5 offspring being sired on the maternal plants there. So what you'll first notice is that most of the points are 0. So a single visit by any of these pollinators did not result in siring on the maternal plants. So what we see is that there is a lot of pollen leaving the system. The other thing I hope that you notice is that Andrena here, Andrena helianthiformis, has the most situations where a single visit resulted in multiple offspring being sired on the maternal plants. I don't have any stats here because I'm still not super happy with the models, but it does seem to be that this is significant. So we can look at the same way we looked at female fitness, where we have male floret shedding pollen on our x-axis and number of offspring sired per pollinator visit on our y-axis. And the first thing you'll notice is that there's really no correlation between the number of male floret shedding pollen, so kind of the male effort on our sire plants, and the number of offspring pollinated on our female plants. Now the points here indicate our different pollinator groups, and what you'll notice again is that our gray dots up here, that's Andrena helianthiformis, and those are in general higher, even though there are a lot down here, while all of the other pollinators are mostly clumped down here. So how I interpret this is that what we have is that we can now conclude that per visit, Andrena is a significantly more effective pollinator than other bees, and we can take away those asterisks, both from a male and female perspective. But then you might be asking, does this matter? So now we know this, but does it matter? So I would argue it does matter. And the reason I would argue it matters is because we did look at the pollinator communities in these different remnant populations. So this is work we published in 2018 in Oikos. These are just some of the remnant populations. The numbers indicate the average number of flowering plants. And the pollinator communities did differ between the remnants. But what key thing for this point is that only the largest remnants here, these 2 remnants, had Andrena helianthiformis visits at all. And most of those visits were in the very large remnant here. And so what we have is we have our most effective pollinator, Andrena helianthiformis, from both a male and female fitness standpoint, is not found in most of the populations, but is only found in these really large remnants. And I think this highlights the fact that in order to preserve plant populations, we need to think about their pollinators and think about what they need to survive as well. So I think this indicates that she needs large populations of echinacea, larger, more continuous populations of echinacea. And with that, I would just again like to thank all the co-authors on this research and my funding sources. And I'm sure I've been taking questions in the chat, but I'm happy to take additional questions. Or if you're watching this asynchronously, you're also welcome to email me or contact me through the Botany portal, and I'm happy to answer any questions. Thank you all. Bye.